चालू कर दीजिए. Hello, friends. Now we'll be talking about the very first topic of biochemistry, that is cell. What is a cell? Got it. Hello, friends. Today we'll be talking about the first basic unit of life, that is cell, which is also the first topic to be discussed in biochemistry. Myself, Dr. Itishri Pandey, and today I'll be getting into the details of cells. In this lecture, we'll be talking about the important university exam questions, since concepts is what you will be getting from the lecture, but then these concepts have to be reproduced in the examination, for which we need to know the frequently asked university questions. The definition of cell, the various cell compartments, cell organelle, and cell fractionation. So the short answers which are most commonly asked under cell is short note on plasma membrane along with the diagram and elaboration on fluid mosaic model. Second one is active transport across cell membrane. Third is cell membrane structure and function. Fourth is short note on ion channel and short note on mitochondria. The long answers question, which is very commonly asked, is describe active transport across cell membrane with suitable example. And the second question is describe cell organelle compartmentalization along with the pathways and the functions they perform. So as we know that our body is made up of different organs. These organs have tissues, and these tissues are made up of various cells. So cell is the structural and the functional unit of life. There are two kinds of living cells. One is prokaryotic cell, which is found in bacteria and viruses, and the second one is eukaryotic, which is found in humans. Now we'll talk about cell compartment. So basically, every cell organelle performs different function, has different set of enzymes. Similar to the distribution at our home, like the food is cooked in the kitchen, there's a different bedroom and there's a different living room. Home is organized to perform different activities. Similarly, cell is compartmentalized or organized into different sections like cytosol, mitochondria, endoplasmic reticulum, lysosomes, peroxisomes, and nucleus to perform different set of functions. Like mitochondria is the powerhouse of the cell because it produces ATP, which is the source of energy in our body. Now let us look at the structure of cell. As we can see that the cell has a plasma membrane. It has endoplasmic reticulum with ribosomes embedded in it called as rough endoplasmic reticulum. There's a big central nucleus along with the nucleolus. There's a cytoplasm, which is a gel-like matrix, mitochondria, microtubules, lysosome for the phagocytosis, smooth endoplasmic reticulum without the ribosomes, and the centrioles. Now let us look at the functions of different organelle. Lysosome is for food digestion, garbage disposal, and recycling. Nucleus being the most predominant organelle, it protects the DNA and controls the cell. Ribosome builds protein. Golgi apparatus finishes, packages, and ships the protein, whereas endoplasmic reticulum helps in finishing the proteins which are synthesized, and it is also helpful in synthesis of the membranes for like lysosomes and peroxisome and other organelle. This is a cell membrane which forms the boundary of the cell. It controls movement and material in and out of the cell, and it recognizes signals because it contains receptors. Mitochondria makes ATP and hence called as the powerhouse of the cell. Centriole is involved in the cell division. Vacuoles and vesicle, it transport inside the cell, and it also used in the storage. Whereas cytoplasm is a jelly-like material which holds the organelles well in place. Now we'll talk about the enzymes and the pathways which happen at various cell compartments. Cytoplasm. The enzymes which are present is aminotransferase and peptidases. The pathway that happens is glycolysis, HMP shunt, fatty acid synthesis, purine and pyrimidine catabolism. Mitochondria is involved in fatty acid oxidation, amino acid oxidation, Krebs cycle, urea synthesis, electron transport chain, and oxidative phosphorylation. Nucleus is involved in transcription and translation, hence the biosynthesis of DNA and RNA, and also replication. Endoplasmic reticulum is involved in protein biosynthesis, triacylglycerol and phospholipid synthesis, steroid synthesis and reduction, and cytochrome P450 and esterases. Lysosome contains phosphatase, phospholipase, hydrolases, proteases, lipases, and nucleases required for the digestion of various macromolecules. Golgi apparatus contains glucose 6-phosphatase, 5' nucleotidase, glucosyl and galactosyl transferase, whereas peroxisome contains catalase, urate oxidase, D-amino acid oxidase, and involved in long-chain fatty acid oxidation. Now we'll discuss about the plasma membrane, in which there is very commonly short note asked, like the structure, the fluid mosaic model, etc. So what is plasma membrane? It is basically a limiting membrane which is required for cellular organization and integrity. Now we'll look at the diagram of the plasma membrane. This is one single layer which is made up of two layers of phospholipid. This is one and this is second. So it is called as biphospholipid or phospholipid bilayer. As you can see here, in this membrane, the proteins and the carbohydrates are embedded. There are two kinds of protein. One is integral protein, whereas the other one is peripheral protein. And there are carbohydrate chains which are present in the plasma membrane. Now the concept of lipid bilayer was given by Davison and Daniel. They stated that the membrane contains glycophospholipid and sphingophospholipid, and it is present as a bilayer. It has a hydrophobic head and a hydrophilic tail. Hydrophobic head is towards outside, whereas hydrophilic tail is faces the hydrophilic tail of the other biophospholipid. Now coming into the fluid mosaic model, which was given by Singer and Nicholson in 1972. It states that the organization of plasma membrane is such that it has a phospholipid bilayer which contains embedded proteins and cholesterol. The molecular arrangement of plasma membrane resembles an ever-moving sea of fluid lipid that contains a mosaic of different protein, and hence the name fluid mosaic model. Some proteins are float freely like tip of the iceberg, whereas others are anchored at specific location like boat at a dock. Now look at the composition of different molecule at cell membrane. So it comprises of 40% of lipid, 60% of protein, and 10% of carbohydrates. In lipids, there is fatty acid, glycerophospholipid, sphingolipids, and cholesterol which is present. This lipid have hydrophobic head and a hydrophilic tail. Fatty acid is the major component of the plasma membrane, which has 50% saturated and 50% unsaturated fatty acid. Unsaturated fatty acid is important for the fluidity of the plasma membrane. Oleic acid is the most abundant fatty acid which is present in the fatty acid in the plasma membrane. Glycerophospholipid, sphingolipid which is present in nervous tissue, and cholesterol which is also important for the fluidity of the plasma membrane. Now we'll talk about the protein. So there are three kinds of protein which are present in cell membrane, namely integral protein, also called as intrinsic membrane, the peripheral membrane or the extrinsic protein, and the third one is transmembrane protein. Integral protein is attached via van der Waal with hydrophobic head, and removing integral protein is cumbersome, whereas peripheral protein gets removed easily from the plasma membrane and it is attached via ionic or hydrogen bond. Transmembrane protein, on the contrary, it spans the whole breadth of the plasma membrane and contains the receptors which is required. Carbohydrate, which comprises 10% of the total, is present in the form of glycolipid or in the form of glycoprotein. Glycolipid is the present as polysaccharide thick coating which is attached covalently to the lipid, and the examples are D-galactose, D-mannose, D-fucose, etc. Now let us look at the diagram here. This is the integral protein which is present across the membrane, peripheral protein which is loosely attached and can be easily removed, and there is glycolipid, various phospholipids and cholesterol which is present in the plasma membrane. Now this image shows the hydrophobic head and the hydrophilic tail of phospholipid. The tail is made up of fatty acid chain, whereas the polar head is attached via phosphate and glycerol. Now we'll talk about the membrane fluidity, which is essential for many functions of the membrane such as permeability and enzyme activity. And the factors which affects the membrane fluidity are temperature, chain length, and the degree of unsaturation of the fatty acid and the presence of cholesterol. So it is the temperature, cholesterol, and unsaturated fatty acid which determines the fluidity of the plasma membrane. Now let us talk about the function of plasma membrane, that it transports molecule across the membrane since it is selectively permeable. It helps in cell-to-cell communication, transmembrane signaling, and cellular regulation. It is required for compartmentalization, and also it is involved in membrane modifications for specialized functions. And last but not the least, it protects the cell. Now there is another organelle which is called as the cytoplasm. It is also called as cytosol. It is a fluid component which contains soluble viscous gel, and it represents 50 to 60% of the total cell volume, and it is in contact with all the subcellular organelle. It contains enzymes, metabolites, and the ions. The enzymes which are present in the cytoplasm has functions in following pathway. In carbohydrate metabolism, it is involved in glycolysis, PPP, glycogen metabolism. PPP is pentose phosphate pathway. In protein metabolism, deamination, transamination, and urea synthesis takes place in the cytosol. In the fat metabolism, fatty acid synthesis, de novo synthesis takes place in the cytoplasm, and also in nucleotide synthesis in purine and pyrimidine. Metabolite, which is produced by the metabolic pathway, is present in the cytoplasm. The ions which are present in the cytoplasm are sodium, potassium, calcium, magnesium, bicarbonate, chloride, and phosphate. Now, this cytosol also contains cytoskeleton, and these are basically the cytoplasmic filaments that is made up of proteins. So these are of three types: actin, microtubules, and the intermediate filament. Actin is required for the cell motility, transportation of material within the cell, whereas microtubule is involved in chromosome separation and it provides cell motility. Intermediate filament, on the contrary, reinforces cell shape and fixes position of the organelle. So in totality, cytoskeleton has role in cell morphology, intracellular transportation, cell motility as cilia and flagella contains microtubules, and cell division because microtubules helps in the formation of mitotic spindle during cell division. Now the applied biochemistry of the cytosol is in a disorder which is called as Carter syndrome, in which the cilia becomes immotile, and this cilia contains microtubules. Now we'll talk about the next organ, which is nucleus. As you can look at the diagram here of nucleus, that the nucleus has two nuclear membrane, outer and inner. It has a nucleoplasm which contains chromatin. In the center there is nucleolus, and nuclear membrane has a pore which is called as nuclear pore. It is a gap between the outer and the inner nuclear membrane. Nucleus, it was first discovered by Robert Brown, and it is the control center of the cell because it contains the DNA, which is organized into chromosomes which carry the genetic information. Nucleus is surrounded by double membrane which is called as nuclear membrane or envelope. The outer membrane is fused with the endoplasmic reticulum at multiple sites. The nuclear pore, as I said, the gap between the two nuclear membrane, which is a multi-protein complexes, occurs at points where the outer and inner membrane are connected. Nucleosome contains DNA and histone. DNA and histone, since DNA is present DNA is present as in combination with histone, forming nucleosome. This nucleosome forms chromatin. The assembly of chromatin fiber forms chromosome. Now, talking about the function of nucleus, as it contains DNA and RNA, it is involved in DNA replication and RNA transcription of DNA. The nucleolus is non-membranes and it contains RNA polymerase, RNase, ATPase, and other enzymes, but it does not contain DNA polymerase. It is also the site of synthesis of ribosomal RNA and assembling of ribosome subunits. Now coming into the disorders of nucleus. Since DNA is present in the nucleus, the disorder of DNA includes disorder of mitosis, apoptosis, chromosomal disorders, and mutations. The disorders of mitosis are cancer, because increased and uncontrolled mitosis leads to cancer. Anti-cancer drugs causes cell arrest and hence used in the treatment of cancer. Chromosomal disorder examples are trisomy 16, 18, and 21. Mutations refers to the change in the sequence of DNA, and hereditary mutations example are cystic fibrosis, hemophilia, and sickle cell disease. Now look at this diagram. This image is from a movie called as Pa. This role was done by Mr. Amitabh Bachchan, in which the child is suffering from a disorder in which the aging precedes at a very young age. So a 2-year, 3-year, or maybe 5-year-old would look like 70-year, 80-year old. Okay, this is called as Hutchinson-Gilford progeria syndrome, and the aging begins at birth. It happens because of the distortion of the nuclear envelope due to accumulation of lemon A. I repeat, in Hutchinson-Gilford progeria syndrome, the aging begins at birth, the distortion of nuclear envelope due to accumulation of lemon A. So this was about nucleus. Now we'll talk about the next organ, which is mitochondria. As you can see in the diagram here, that mitochondria has outer and inner membrane. It has its own set of DNA. It has a matrix, the folded cristae, ribosomes, and granules. Mitochondria is bounded by two concentric membranes that have different properties and biological functions. As I've said that there is outer mitochondrial membrane which consists mainly of phospholipid, cholesterol and proteins. The inner mitochondrial membrane which is rich in proteins, and the inner membrane projects inwards into fold that are called as cristae. And the intermembrane space between the outer and the inner nucleotide, outer and inner mitochondrial membrane, is involved in nucleotide metabolism. The mitosol or the gel-like matrix consists of enzyme required for the pathway like glycolysis, oxidation of pyruvate, etc. And it is also the site of the reactions of the TCA and fatty acid oxidation. The components of electron transport chain and oxidative phosphorylation, which is responsible for the synthesis of ATP, are embedded in the inner membrane. As I have shown in the first picture that mitochondria contains mitochondrial DNA, which in human encodes 13 respiratory chain proteins, which are small and large ribosomal RNAs and enough tRNAs to translate all the codons. Mitochondria have a key role in aging, and cytochrome can be an initiator of apoptosis, which is present in mitochondria. Now let us look at the clinical disorder which is related to mitochondria. Mitochondrial DNA can be damaged by free radical, and age-related degenerative disorders like Parkinson's disease and cardiomyopathy. Mutational defect that leads to mitochondrial myopathies are Leber's hereditary optic neuropathy, abbreviated as LHON, mitochondrial encephalomyopathy, lactic acidosis, and stroke-like syndrome, abbreviated as MELAS. Third one is myoclonus epilepsy with ragged red fiber, abbreviated as MERRF. These are the clinical disorders related to mitochondria. There are inhibitors which are cyanide and carbon monoxide. They inhibit cytochrome oxidase, which is a component of the electron transport chain and thus decreasing ATP synthesis. Hence, they act as respiratory poisons. Megamitochondria, as the name suggests, larger mitochondria, is seen in viral hepatitis and alcoholic liver disease. Now, the next organelle is Golgi apparatus. It was given by Camillo Golgi, described the structure in 1898, and it is a network of flattened smooth membranes and vesicles, as we can see in the image here. In Golgi apparatus, proteins are processed, modified, and prepared for the export from the cell. It works in association with endoplasmic reticulum, where proteins for certain destinations are synthesized. It is a secretory cell organelle. It synthesizes protein and products and stores it and releases under appropriate stimuli. Example is pancreatic digestive enzyme like trypsinogen. Membrane, it is also involved in the membrane synthesis of peroxisomes and lysosomes. The modified proteins are sorted, packaged, and transported to destination inside or outside the cell from the Golgi apparatus. Post-translation modification of protein occurs in the lumen. Golgi bodies and endoplasmic reticulum give rise to primary lysosomes and the peroxisome, and the acrosome of the sperm cell is derived from Golgi bodies. Now, the disorder which is related to this organ is eye cell disease, which is also called as inclusion cell disease or mucolipidosis 2. It is a disorder of Golgi function which is caused by impairment in the delivery of enzyme which is destined for the lysosome. So, this disorder is also the disorder of lysosomes. The next organelle is ribosome. बस उठा ही लेते, चाय के लिए बोल रही थी, बस रिकॉर्डिंग नहीं हुआ, 15 मिनट और है। हाँ, चाय बस बोल देना। Ribosomes. Ribosomes, they are made of proteins and ribosomal RNA, and they are either attached to rough endoplasmic reticulum or they float freely in the cytoplasm. In eukaryotes, ribosomes are 80S, which consist of 60S large subunit and 40S small subunit. The largest subunit, that is 60S unit, contains 28S, 5S, and 5.8S ribosomal RNA. Small subunit contains 18S ribosomal RNA. Ribosome is also the site of protein synthesis. Endoplasmic reticulum, as you can see in the image here, either it is smooth endoplasmic reticulum without ribosome, or it could be rough endoplasmic reticulum to which ribosomes are attached. It has a cisternae Endoplasmic reticulum, it is a network of interconnecting membranes which encloses channels or cisternae that are continuous from the outer nuclear envelope to outer plasma membrane. As I have said earlier, rough endoplasmic reticulum is endoplasmic reticulum with the ribosome. Microsomes is disrupted rough endoplasmic reticulum that forms small vesicle. Now let us talk about the functions of rough and smooth endoplasmic reticulum. Rough endoplasmic reticulum synthesizes membrane lipids and secretory protein, and these proteins are inserted through the endoplasmic reticular membrane into the lumen of the cisternae, where they are modified and transported to the cell. Whereas smooth endoplasmic reticulum is site of synthesis of phospholipids, cholesterol, and steroid hormones. It is also involved in glycogen metabolism in liver and muscle. It is also involved in the storage or release of calcium in the cell, that is sarcoplasmic reticulum of the skeletal muscles. It is also the site of detoxification of drugs and chemicals in liver and kidney. The next organelle that we will be discussing is lysosome. It contains packet of enzymes, and it is also called as the digestive tract of the cell because it is involved in digestion of macromolecules like proteins, lipids, carbohydrates, and nucleic acid. The lysosomal enzymes have an optimum pH around 5. Let us look at the structure of the lysosome. In the center, there is hydrolytic enzyme mixture which is present, outer membrane, and which is attached to lipid bilayer, and the membrane transfer proteins which is embedded into it. The enzymes which are present in the lysosomes are as follows. First is polysaccharide hydrolysing enzymes, which are alpha glucosidase, fucosidase, etc. And protein hydrolyzing enzymes are cathepsins, collagenase, elastase, and peptidases. Nucleic acid hydrolyzing enzymes are ribonucleases and deoxyribonucleases. Lysosomal enzymes destroys the foreign substance and pathogens like bacteria, viruses that are engulfed by the cells of the immune system. Thank you. Lysosomal enzymes destroys the foreign substances and pathogens like bacteria, viruses that are engulfed by the cells of the immune system, destroying aging cell organelles that are no longer required by the host cell. They are also capable of self-destruction of the cell called as autophagy. Lysosomes are called as suicide bags and plays an important role in apoptosis, and after the death they play a role in post-mortem autolysis. Now talking about the process of digestion that takes place in the lysosome, it is via two processes. One is phagocytosis and the other one is endocytosis. So in the process of phagocytosis, the foreign particle gets engulfed by the phagosomes. The foreign particle enters to form phagosome, and this phagosome fuses with primary lysosome to form phagolysosome or secondary lysosome, which gets further degraded. In the process of endocytosis, endosome is formed, which gets fused with primary lysosome to form endolysosome, which gets degraded further. So this was the process of digestion in the lysosome. The applied part of it is that lysosome is involved in the gout, postmortem autolysis, as we discussed. Cathepsin is involved in tumor metastasis. It also has a role in silicosis and eye cell disease, as we have discussed previously. The next organ is now the last organ is peroxisome. It is also known as microbodies, and it is a single membrane cellular cell structure. These vesicles are concerned with the metabolism of hydrogen peroxides, and hence the name peroxisomes. They contain such enzyme that forms, uses and destroys hydrogen peroxide, and the hydrogen peroxide which is produced in the cell are highly toxic and is decomposed into water and oxygen by the enzyme catalase which is present in them. The function of peroxisome, as we have discussed, it causes breakdown of hydrogen peroxidase. The function of peroxidase, as we have discussed, it contains catalase which causes breakdown of hydrogen peroxide, which is a free radical. And they also help in the synthesis of glycolipid, plasmalogens, isoprenoids and bile. Now the clinical condition which is associated with the peroxisome is Zellweger syndrome, which is also called as cerebrohepatorenal syndrome. Remember this syndrome, it is most commonly asked MCQ in the university examination, that the organ which is involved is peroxisome, and there is accumulation of long chain polyunsaturated fatty acid. I repeat, in Zellweger, there is accumulation of long chain polyunsaturated fatty acid in the brain, liver, and kidney. So basically, it is an inborn error of peroxisomal dysfunction in which there is absence of the functional peroxisome, and other biochemical findings are low tissue plasmalogen level, and as I have said earlier, there is accumulation of long chain polyunsaturated fatty acid. Clinical features are neurologic and hepatic dysfunction, and victims usually die within 1 year. The victims die within 1 year after birth. The other disorders which are related to peroxisome is adrenoleukodystrophy and Refsum disease. Now, when we talk about cell, there are various cell organelle which is present. So the important part about cell is that whenever we have to understand a particular disorder or any function, we have to know the underlying pathology which is at the molecular level, and to study that we need to separate various cell organelle. So the cell fractionation is done using ultracentrifuge. The steps involved are extraction and homogenization, centrifugation, density gradient, and centrifugation. Now, the whole cell separation is done via purification of cell on the basis of density, or in flow cytometry, which separates different kinds of cell like eosinophil, monocytes, etc. Okay, so this was about the biochemistry of the cell, the different cell organelle, their function, their structure, and the disorders which are related to that particular cell organelle, and how we separate this cell organelle to study the biochemical changes is via using ultracentrifuge. So this was all about cell. Thank you so much. Oh. हो गया